Good morning. How are you all? Are you getting any benefit from the YouTube link? Have anyone visited? Can you share your experience? Just unmute yourself and respond. Sadia Afrin Tabassum, are you in the class? Sadia Afrin Tabassum?
Yes, sir.
Have you visited the YouTube link to get more insight or repeat revision of my lecture?
No, sir.
Not yet. Okay, some of you of the students, they sent me email and responded their feedback, how they are feeling my lecture as well as their impression. And most of them are very good feeling. This is really good. The feedback is very important. I have been a adjunct faculty member of East-West University. They hired me for genetic engineering and biotechnology department, and I worked there part-time from 2014 to 2017. What I found the difference between public university and private university, most of the students over there, they are powerful in communication. So they have the system to judge the teacher's performance at the end of the lecture, and otherwise they would not get the result. And in that case, they have everything online. In that case, out of 5 scale, I always record at the top, and they also add the students comment. But in our university, a public university, and Bangabandhu is a unique university, our students are brilliant enough, almost homogeneous at their background. But most cases, they feel shy to ask questions. But when you go in your real life, you cannot keep you silent. If you are silent, nobody know you, and they have no time to learn about you. So the best idea to prepare yourself in a well communicated personality, and one of the way is to interact with your teachers in the class. And I have given you another opportunity to ask question through the Facebook. So in your Facebook is a virtual classroom, which is asynchronous. Anytime from anywhere you can ask question. So my suggestion today, please ask at least one question and share at least one relevant link by browsing the Google before next class. I want to see your skill of communication. And some of the students already sent me mail, and I found them they are really efficient in communication. There are some weaknesses because in most of the cases I found in their mail, they addressed very nicely and wrote English very clearly, but no signature. And you need to close your email and put your contact details, and it is very important for the academic or professional communication. Anyway, today we shall discuss or continue our discussion on the structure functions of DNA RNA, and why the structure functions as well as the expression of the gene and other things important for our life. And do you have any experience? So I shall share with you. By the way, have uh, anyone joined the orientation program because it was virtual? Maybe new students only, not right?
Sir.
Okay. Anyway, I'm sharing the slides. So is the slide visible to you?
Yes, sir.
Very good. In our earlier lecture, we discussed about genetic code and different combination of 4 letters or 4 bases, how they make 3 letters code. Genetic code is basically a triplet in nature. That means in the RNA, adenine, guanine, cytosine, and uracil are present. So these 4 letters, from these 4 letters, how many combination of 3 letters can be possible? And it was our previous discussion. And here is the different genetic codes in a tabular form. For example, uracil, and uracil, and it recruits phenylalanine. So when messenger RNA carries the information, each 4 letters instruct for recruiting a particular amino acids, and tRNA in the ribosome does the job. And finally, a protein is synthesized, and protein is a polymer of amino acids. So genetic code, they have codes have some properties. First property is triplet in nature. Second, non-overlapping. Third, non-punctuating, no space or no punctuation. And fourth, degeneracy, that means synonyms. And fifth, non-ambiguous. Six, universal. And 7, oblique. Now question is, what is degeneracy? Degeneracy means one amino acid is coded by many number of codons. It is a degeneracy, that is several codon can recruit or carries the message of one amino acid. And another is point I mentioned at the point 7, oblique. Oblique hypothesis. There is a hypothesis. The specificity of the codon is determined by first 2 bases. This is very interesting. There are 3 bases in the codon, but 2 bases determine which amino acid would be recruited. This is very important. And these 2, degeneracy and oblique hypothesis, are linked, because I mentioned even same code, different amino acid may be synthesized or recruited because 2 bases, first 2 bases determine which one. This is another point. There are, you have seen huge number. Huge number of codons, and among them 3 codon or code you can see UAA, UAG, red mark, UGA, they are stop codon. They are not involved in recruiting the amino acid. And non-punctuating means genetic code is non-punctuating, as I mentioned. The reading frame of mRNA code not have any break during the translation. It is a sequence. mRNA has a linear, a single-stranded sequence. So there is no punctuation and there is no break for translation. Punctuation in codon may be lethal. If there is any space, any punctuation, or any, discrepancy, then it may be lethal. Lethal means it is problematic for the translation. It is called as a comma-less language. That means DNA RNA is a comma-less language, no punctuation actually. There are some important terms I should share with you. The first term is genome. This is very important term. What is genome? The entire genetic material, DNA and RNA of an organism is called genome. That means, for example, oryzae sativa, rice. Rice has a genome. So in the rice, the DNA present in the nucleus, mitochondria, and plastic. So entire genetic material is called the genome. The Jude genome sequence has been done by Professor Maksudul Alam, a brilliant professor who was my good friend, and he received the our Independence Day award, and he died prematurely, around 60 years, unfortunately. He is famous for Jude genome sequence. That means decoding of the genome sequence means a genome sequence is the complete list of the nucleotide. For example, A, C, G, T for the DNA genome, or RNA genome, it should be A, C, G, U that make up all the chromosomes of an individual or a species. Even nowadays, the genome sequence of SARS-CoV-2 is very popular because Bangladesh also did or sequenced several hundreds of virus SARS-CoV-2. And by doing genome sequence, in case of COVID-19, what we can understand, where the sequence has been changed. We called it is some variant or like that. Now, Omicron variant is going on. And when genome sequence, in the genome sequence, slight change may change the character of the organism. And you vastly know about the SARS-CoV-2, severe acute respiratory syndrome, coronavirus 2, which causes COVID-19 disease. Transcriptome, another term, like genome sequence. Transcriptome, transcription. Transcription, when DNA transcribed to mRNA, then we can do also sequence of mRNA. The transcriptome is the set of all RNA molecules in one cell or a population of the cell. And it is sometimes used to refer to all RNAs or just mRNA, depending on the particular experiment. So transcriptome is also very important because DNA, a certain amount of DNA always transcribed to mRNA. So if you sequence the mRNA, extract the mRNA from the cell, and if you sequence the RNA from the cell, then you can get the information of what are the genes how to say, transcribed or expressed. mRNA is very, very important. By sequencing mRNA, you can get lots of information about the activity of the DNA. That means the genetic properties of the expressed genetic properties of the organism. So this is another subject, transcriptomics. And where the other term is proteome. The proteome is the entire set of proteins that is or can be expressed by a genome, cell, tissue, organism at a certain time. It is the set of expressed proteins in a given type of cell or organism. So it is clear that first genome, and then transcriptome, because genome is entire genetic material, for example, DNA. And when DNA is transcribed into RNA, the entire RNA in the cell we can call transcriptome. And when transcriptome translate, mRNA translate, then it synthesizes the protein. So in a given time in the cell, the entire set of proteins, lots of proteins are, they are doing the job due to the expression of gene. They are called proteome. Interesting is that mathematically they are connected. So by using computer software, if you know the proteome, then you can know the relevant mRNA, and then you can know the relevant DNA that expressed or transcribed. The last one is metabolome. The metabolome refers to the complete set of all molecule chemicals found within a biological sample. That means when protein is synthesized, some of the proteins are enzymes. They participate in different metabolic pathway, for example, TCA cycle or glycolysis or other. There are lots of metabolic pathway to synthesize different metabolite. Metabolite is the product or intermediate of metabolism. In the cell, there are two types of metabolism. One is anabolism, another is catabolism. So in anabolism or catabolism, in both cases, metabolites are very important. So metabolome means entire set of small molecule like, for example, carboxylic acids, acetic acid or like that, or an alkaloid or others. So if you know the metabolome cannot synthesize of its own. It needs synthetic pathway, biosynthetic pathway, and that pathway is governed by different enzymes, because enzymes are the catalyst to transform one metabolome to another, as you have seen in the glycolytic pathway or TCA cycle. So from the metabolome, you can, by using bioinformatics software, you can get the information of protein, which protein means enzyme is involved in the synthesis of that metabolome. From the proteome, you can go for the transcriptome. From the transcriptome, you can go to the DNA. So these are the important subject to dissect the molecular mechanism of any phenotype we can see in the organisms we can reveal. One interesting bank is genebank. The bank where we deposit money or get money from our account or take loan or lots of other things. Similarly, genebank is very famous in the molecular biology. Gene banks are a type of biorepository which preserve genetic material or sequence data of genetic material or biomolecules. For example, NCBI Gene Bank, National Center for Biological Information. So it is owned by the USA. There are several other gene bank. Japan has own gene bank. Europe has their own gene bank, but Bangladesh has not yet any gene bank. So genomic data, for example, DNA, RNA, or proteomic data or metabolomic data. When you get the sequence of any from any organism, you can deposit there. And the good point is that globally, people share it freely and deposit in the usually share it freely in the deposit in the gene bank. For example, Jute Genome Project or Hilsha Genome Project or Goat Genome Project, the sequence data have been deposited in the gene bank. So if you browse, you can find it. And every day, thousands of data are deposited in the gene bank, for example, NCBI. And all the gene banks are connected. So they are browsable. And really amazing mass of knowledge are deposited. For example, in case of coronavirus, gene bank is very important. Coronavirus, to tackle the coronavirus COVID-19 disease, scientists decided to deposit all of their data in a special gene bank, GIS AID. So all the data, how to say, millions of sequences of coronavirus SARS-CoV-2 from all over the world are deposited over there, and anyone from that sequence can see and utilize them for developing diagnostic protocol. For example, the PCR RT-PCR method. So to develop the RT-PCR method, the sequence of RNA of SARS-CoV-2 virus was very important, because from that sequence, you can pick up the unique sequence which has not been found in any other viruses or other organisms. You can use that sequence as a marker and identify precisely that SARS-CoV-2. So genome sequence, or all the sequences, genome, transcriptome, proteome, or metabolome sequences, are very, very important and relevant for practical application. And from the genome sequence of coronavirus, vaccines are designed, and different therapeutic interventions are also developed, and even we could identify the variants, new variants and others. For plants, this could be by in vitro storage gene bank. There are other gene bank, for example, in vitro storage of or freezing of some plant, or stocking the seeds. There are seed bank also, but gene bank is different. Gene bank is associated with the sequence data, sequence of DNA, RNA, sequence of protein. If you sequence what you would get, amino acid, the sequence of amino acid, and you can also get the information about metabolome, and you can submit in the gene bank, and gene bank will give you an accession number like your ID, unique number. Plant genetic material in a gene bank is preserved at minus 196 degrees Celsius in liquid nitrogen. This is also one of the gene bank, but these genes are not defined. Even sperm can be deposited in the gene bank, and they are not, a cell. So they are cell basically, not the pure DNA or RNA. And minus 196 degrees Celsius means cryopreservation. So there is a term called cryopreservation. Cryopreservation means a living cell or tissues, if you preserve at minus 196 degrees Celsius, it is called cryopreservation. In our lab, we have minus 86, not minus 196. And minus 86, we can preserve the bacteria for longer time, fungi. But if you want to preserve, for example, human sperm, then you need minus 196 degrees Celsius. Then sperm will survive even after 200 years. You can utilize them. So to handle all the data, that is DNA sequence, RNA sequence, or protein sequence, there is a new subject called bioinformatics. Bioinformatics means bioinformatics is one kind of statistical analysis, but in case of sequence, millions of data are generated, so normal statistics cannot do it. The body of tools, algorithms needed to handle large and complex biological information is called bioinformatics. That means bioinformatics is some sort of software we use to determine the information in the data. And computer scientists, they use the algorithm to design the software. And this is very important in the biotechnology. It helps manage molecular biological data, store in databases, organize, formalize, and describe, compare molecular biological data with one another. For example, you have a sequence data, genome sequence data of basmati rice, and you have another genome sequence data of Kalizira rice. Then you can compare both genome sequence data by using software and find what are the uniqueness of basmati and Kalizira. Then you can understand why they are different, although both are oryzae sativa. Find patterns in molecular biological data, that is phylogenies. This is very important. Phylogeny. Phylogeny means, for example, if your DNA sequence is done, and if we use the computer software and search the gene bank, and then all the members, your generation deviated all over the world in last, for example, 5,000 years. The relationship you can find. In that case, other members should have the sequence in the gene bank. For example, if your family members, all of them have sequence data, then you can find the relatedness of the genome sequence among the members. And also correlation, sequence, structure, expression, and function. Phylogenomic sequence, for example, you discovered a fungus, then it is which fungus. By using phylogenetic analysis, you can clearly understand which species is it and how it is related to the other fungi in this earth. For example, SARS-CoV-2. When SARS-CoV-2 sequence was done, then it was, phylogenetically analyzed, and it was found that it is a beta coronavirus, and it is different from, closely related to SARS virus, previously SARS, severe acute respiratory syndrome coronavirus. That means it is a coronavirus. You can determine like that way. So the bioinformatics is the subject which we use to characterize biological patterns and processes in the biological data, complex biological data, and predict biological properties by analyzing the sequence by using software. You can predict the genes, you can identify the gene, and you can predict the function of the gene. So this is really important. I'm now telling you one of the exciting story which I experienced in Bangladesh in 2016. That was a horrible, we have now a pandemic. That was an epidemic outbreak of a plant disease called wheat blast, which devastated 15,000 hectares of our cultivated wheat in February. And February, the flowering time of wheat. So government yield loss was up to 100%. Here in the Bangladesh map, you can see 8 districts were devastated, and 15,000 hectares in case of Bangladesh is big, amount of wheat land. 15% of the wheat yield was decreased in the country at that year. And you can see the fire, because as we are not familiar to that disease, and it was like SARS-CoV-2. So government decided to burn the crop because there were no grain, and to reduce the fungus or like that. As I am a student of biotechnology, it makes huge temptation to me to understand what is it. Then you can see in the field how wheat blast looks like. All the spikes are white head symptom, but plants are green. What happened, as you are a student of agriculture, you can understand the fungus at the base of the spike that is at the neck, it infects and block the vascular system. So water transport was blocked, and ultimately the spike dried up. If you apply the fungicide, you can kill the fungus, but already loss was done. And this is the fire in the BADC farm, and it triggered me to do something. The first question was, what is the enemy? Because in our textbook, this disease was not described, and in Asia, it has this type of disease have never been. So I utilized RNA sequence analysis. That means collected the sample from 8 districts, and then in collaboration with international scientists, because I have been in abroad for more than 10 years, I have lots of international big giant researchers. So by using Twitter and Facebook, I collaborated with 31 scientists from 4 continents, and we together applied the genome sequencing, genomics, not going to isolate like plant pathologists do and take lots of time. So we extracted the DNA from the infected plant tissue, not exactly DNA. We extracted the RNA, as I told you, when DNA translate into RNA. RNA is the real matter, and you can get lots of information from the RNA. Our target was to understand the source and origin of the pathogen, whether it is somehow due to climate change mutated in the native fungus and became the pest of wheat, or it came from South America, because this pathogen has been in South America since 1985. Only one continent, Brazil, Argentina, Bolivia, and Paraguay, but all other countries in the world are free from this dangerous pathogen. By using, Trinity software and other software, we pinpointed the disease as Magnaporthe oryzae, and you see, this is called Phylogenetic Tree. Here, our isolate closely matched with the isolate of the South American, Brazilian one. Directly, this is all the isolates in the gene bank. They are originated from the Brazil. So you see our 12,627 number of isolates we collected from the field. They fit well with the Brazilian isolate. That means the origin of our fungus is from Brazil. And now question is, from Brazil, how they came? They came through the rotten wheat grain imported in 2015, and they came through passed through the quarantine because there were no detection method. I shall discuss you later on. But what I am telling you, we precisely confirmed the origin of the fungus that is South America. And what is the benefit of that discovery? That the benefit is, then we asked South America in last 35 years, how they are fighting and what agricultural technologies they developed. And we then applied those technologies in our field to suggest to tackle the, next time epidemic, and there are no epidemics. So these discoveries helped us to save millions tons of wheat in our country. And it was a big news. You can see the in the newspaper, Protomalu, Gomer Shotru Shonakto. It was really amazing news, and you cannot find any discovery in agriculture or any science covered the 3 column of the first phase page, and it was unprecedented. And it was also news in the weekly Nature and Science. And finally, we published the paper in the world leading very famous journal, BMC Biology. So by using genomic technology, that means sequencing technology, Bangabandhu Sheikh Mujibur Rahman Agricultural University made a big breakthrough and helped the nation. And I led the project. Here you can see 31 researchers. I was the first author and corresponding author. And we are working a lot. Even some of your friends attending this class, they showed high interest in joining our mission to mitigate the wheat blast. We are applying gene editing, CRISPR-Cas. We are applying gene integration. We are applying mutation breeding. We are applying many biological or biotechnological approaches to mitigate this problem, because this problem is a big problem for our food security, and it will move to India and China. Maybe it takes time, but India is the second highest wheat producing country in the world, and China is #1. And if this terrible fungus moved there, then it would be catastrophic. So before that, my team is working hard to discover all the knowledge so that we can solve the global problem. And in the world, our team is #1 in wheat blast research. We have 2 postdoc, 2 PhD, and several master's students, even undergrad students. One interesting point I would like to discuss with you briefly, that is, there are debates on GMO, genetically modified organism. But GMO can be produced naturally. Many people do not know. One of the important points is that when scientists first sequenced the whole genome DNA of a human, they found that 33% of our DNA sequence are not originated from the primate or animal. They came from the microorganisms, because an adult human carries 3 to 6 pounds of microorganisms belonging, 150 species. So over the years of carrying the microorganism, even in your body, you are using shampoo or soap every day and taking the bath, but you are carrying 3 to 6 pounds of microorganisms, and they are very beneficial for us. 99.99% are helping us to digest food and protect ourselves from the diseases. But anyway, human is also natural GMO. There are many natural GMO has been discovered by scientists. That means genetic modification taken place through the horizontal gene transfer. For example, the striga. Striga is a parasitic plant for the sorghum, and striga infect the sorghum plant. And over the years of this association, antagonistic association, some genes from the striga move to the sorghum. That is, genetic engineering is done by nature, and sorghum is a natural GMO scientist has discovered. So you can nothing to do. So GMO, the debate is actually meaningless, because when we release a GMO, for example, BT brinjal in Bangladesh, first GMO crop, lots of biosafety assessment have been done, and BT gene in last 40 years didn't harm to the environment or the human. So GMO is the important breakthrough in biotechnology. So you can modify a plant as you desire. And the latest, genetic engineering technology is CRISPR-Cas genome editing. Homo sapiens, also a natural GMO, as I mentioned, about 33% of human genome contains genes from the trivial microorganisms, and adult human carries 3 to 6 pounds of microorganisms. Total number of microorganisms in human body is more than 100 trillion, even higher number than the total number of cells you have, but you cannot see them because they are microorganisms. You need microscope to see them. And there are about 150 species. I would like to share a few information before stop my lecture. The first genome of a plant was done, and its name is Arabidopsis thaliana. It is like a mustard-like wild plant. Why we did genome sequence of this plant for the first time? Because its genome size is small and it has several other advantages. Life cycle is short, and this is called a model plant for the understanding the biology of plant, and its size is only 135 mega base pair. There are human genome project being completed in 2003, and it was a historical project, very expensive project. So it was the cover story of the nature, and many people are now working on dissecting the function of each gene, so that lots of information every day is generated, so that we can understand the decode all the genes and understand the mystery of life in the DNA, so that we can use them in the modern treatment and solve lots of problems. How gene works? This is an amazing picture. You can see a patch in the baby and patch in the mouse. Both are due to the mutation of the same gene. It is called KIT gene. That means how gene regulates the phenotype, that is external properties you can see here. And as a human genome, DNA is, I mentioned you, if you put all the nucleotides one by one and put 1 millimeter space among the nucleotides in the human DNA, it would be 3,200 kilometers or approximately 2,000 lines, 2,000 miles, that you can understand far enough to stress across the center of Africa. You can understand how very long is the DNA sequences. I will give you a gift of this book, Molecular Biology of the Cell, 5th edition. I have the soft copy, and this is just like a Bible in biotechnology or molecular biology. This is all about my today's talk. Now it is your time to ask some question.
Sir, what is the difference between genomics and bioinformatics?
Yes, not a difference. Actually, genomics, omics technologies are very important in the modern science. For example, genome, entire genetic material of your DNA, your cell is called genome, your genome. So you can do the sequence by using machine. In our lab, we have Illumina sequencing, world latest sequencing platform. You can have the sequence data. As I mentioned, if you put all of your nucleotide one by one with 1 millimeter space, then it would be 2,000 miles. So how can you understand the sequence? By using bioinformatics, the software, you can analyze your sequence, and it is called genomics. Analysis of the genome is called genomics. And how can you do the analysis? By using bioinformatics software. And bioinformatics means application of computational methods like algorithm to understand the biological data like genome or proteome or transcriptome is called bioinformatics. So it is a branch of science.
Thank you, sir.
A very nice question, honestly. And any other?
স্যার ওবিং এর বিষয়টা আরেকবার বলে ভালো হয়।
Hmm?
স্যার স্যার কোড প্রপার্টিস এর ভিতরে একটা পয়েন্ট ছিল 7 নাম্বার পয়েন্টটা,
Yes.
স্যার ওটা নিয়ে ডিসকাস করলে ভালো হয় ওবিং।
I told you that. Let me show you. Can anyone share this point you followed nicely? Ubing. Can anyone share, because whether you are following my lecture is very important. Can anyone share this point?
Sir, assalamu alaikum.
Yes, walaikum salam. What should be the answer of the oblique?
Amino acids specificity determined হয় হচ্ছে first two base দ্বারা, এটাকে
Excellent, because genetic code is 3 letters, but which amino acid 3 letters is associated with the information of which amino acid will be recruited during protein synthesis. So first 2 letters determine which amino acid. So it is called oblique like that. Excellent. Shorna, right?
না স্যার লামিন।
Lamin, brilliant, brilliant. Any other question?
Assalamu alaikum, sir.
Nazmus?
Sir, Rokibul.
Rockable, okay. Yes, rockable, yes.
Sir, এখন বাংলাদেশে স্যার হুইট ব্লাস্টের অবস্থাটা কি?
Nice question. Wheat blast, we suggested the farmers not to cultivate wheat in the hot spot, that is Meherpur, Chuadanga, Joshur, the districts that were devastated because fungal, inoculum over there are severe. And they unfortunately they are cultivating now tobacco, because tobacco company, British tobacco company, took the advantages. And we suggested lots of agronomic and disease management approaches that have already been discovered by the Brazilian and other South American countries. But the problem is there is no clear-cut solution for this disease being discovered. There were no diagnostic protocol like RT-PCR or smart technique. We for the first time discovered the diagnostic protocol. Wheat blast moved to Zambia, also African countries, because wheat is #1 export import crop. So our technique, like a pregnancy strip, by using CRISPR-Cas technology, for the first time we discovered, and it is now being used worldwide. This is one approach. Second approach is our Bangladesh Rice Agriculture Research Institute that developed a zinc-rich variety, Barigom 33, and fortunately that variety is 70% resistant to wheat blast. It carries 2NS, some genetic resistant gene, but not a solution, because same gene in the South America been override by their local fungus. So this fungus is terrible, very quickly mutate and resistance develop. Fungicide does not work. So our team is developing gene edited wheat, gene integrates new wheat variety. If you are in the campus, you could see our students how they are working day and night in the campus, our field, as well as in the Meherpur. Hopefully, by a couple of years, we can give the solution. But we developed biocontrol bacteria that can, if you spray, you can control the disease. We developed nanomaterials. If you spray, then you can control the disease. So our team is now #1 in wheat blast management, but real durable management of wheat blast is yet to be done. We need to work more. And if you have, we have the solutions, then we will be world leaders and solve the problems in other countries. Thank you so much, Rokibul.
স্যার আমাদের স্লাইডগুলো দিলে একটু ভালো হতো স্যার।
Yes, of course. I shall provide you the slides, but I advise you to visit my YouTube channel because I spend lots of time to offer you the, the live lecture so that you can browse as many times as you need and try to do that. And please make your comments on the lecture because it may help me to give you better how to say delivery or training or guidance for learning the biotechnology. We have only one course. It is stressful for you. So whatever you feel, please make a comment. And you must ask one question through the Facebook, at least one question, and share at least one content related to our lecture to make the Facebook group an interactive and engaged platform, virtual classroom. Any more question? If no question, then I should thank all of you. Yes.
স্যার আপনার ইউটিউব চ্যানেলটা ভিজিট করেছি স্যার, সেখানে মেবি আমাদের ফার্স্ট লেকচারটা আপলোড করা হয়নি, সেকেন্ড লেকচারটা দেখেছিলাম, তো ওটা কি আপলোড করা হবে?
Yes, of course, first lecture should be there. And I already shared through your Facebook group, and you can find it. And even you can find the lectures that I delivered in the previous years on the same course. So you can browse over there and you can find it. And if you do not find, please contact with me. I shall give you the link. Thank you so much. And I shall obviously give you the lecture slides in PDF format so that you can follow the slides also. But slides are not enough because I talk too much outside the slides, which is very important for you understand the course. Our question would be, innovative, and if you do not follow the lecture thoroughly, you cannot, understand well. And understand, based on understanding, you can get all of the students can get A plus, because in my life I never stood second at BAU. I know how to give the marks, so I can give you 100 out of 100. I don't care, but the problem is your understanding should be crystal clear. So try to understand the course and you can get A plus surely. And if you 100% of the students you earn the A plus, I would not mind. I will be very happy. Thank you so much.